When we talk about memory, you probably think of what's happening in your brain, right? But today we're going way, way deeper than that. We're talking about the chemistry of memory itself, how non-living chemicals first figured out how to remember and how to copy themselves. This is the incredible scientific detective story of how life itself might have gotten its start. So let's jump right in.
I mean, this is the big one, isn't it? How did life begin? You look around at all this incredible complexity, this biological machinery that replicates itself, and you have to wonder, how could all of this possibly come from just a bunch of simple, lifeless chemicals? It almost feels like it shouldn't be possible.
Well, this investigation has been going on for a long time. Our first big break in the case came way back in 1953. But the story itself? Oh, it starts 4 billion years ago, on a very, very different and chaotic early Earth.
Okay, so let's start our story with that first major piece of evidence. The first big clue that suggested the building blocks of life weren't some kind of miracle, but something that could just happen naturally.
So back in 1953, two scientists, Miller and Urey, did something pretty amazing. They basically built a tiny, primitive Earth in a flask. They put in the stuff they thought was in the atmosphere back then, water, methane, ammonia, hydrogen, and then they zapped it with electric sparks, kind of like mini lightning strikes.
And you know what? It worked. What they found was just mind-blowing. Amino acids, the literal building blocks of proteins, just formed all on their own. But, and this is a huge but, there was something really important missing. They found no DNA, no RNA, absolutely nothing that could actually store the instructions for life.
So that missing piece, it led scientists straight into a brick wall. A huge, frustrating paradox, a classic chicken and egg problem that basically stopped the whole investigation cold for years.
Okay, so here's the problem in a nutshell. Think about it. In all living things today, DNA and RNA are like the blueprints. They hold all the instructions for building everything. But you need proteins, these special little machines called enzymes, to actually read the blueprints and copy them. So what came first, the blueprint or the machine that reads the blueprint? It's a total catch-22. You absolutely need one to make the other.
And for years, that was the big roadblock. Nobody could figure it out. But then, in the 1980s, a brand new theory popped up, a new lead in the case that offered a really clever way out of this mess. Enter the RNA World Hypothesis. The idea is, what if, way back in the beginning, there was a single molecule that could do both jobs? And it turns out RNA is a pretty amazing molecule. Scientists found that not only can it store information, kind of like DNA, but it can also fold up and act like a little chemical machine, an enzyme. It was the perfect suspect to break that chicken and egg cycle.
Okay, so problem solved, right? Well, not quite. There was another big catch. Scientists got pretty good at making RNA enzymes that could build other RNA molecules, but they couldn't for the life of them find one that could copy itself. The ones they made were just too big, too complicated to fold into the right shape to work as a machine. They were too complex to also work as a simple template. So, another dead end.
And that brings us right up to the present day, and to the big breakthrough. Because just recently, a team of scientists in the UK finally did it. They found the thing everyone has been searching for decades, a molecule that could finally, actually copy itself.
They called it QT45. And the secret, the real magic, was its size. It's tiny, only 45 nucleotides long. That's way, way smaller than anything people had tried before. It turns out, for this puzzle, the answer wasn't more complexity, it was simplicity. And the way it works is just beautiful. It's so elegant. First, the molecule builds a negative version of itself, kind of like a photographic negative. Then, it uses that negative as a mold, a template, to pop out a brand new identical copy of the original. Boom! That's it. Self-replication solved. But this is where the story gets really, really interesting, because it turns out this little molecule is, well, it's not perfect. It has some pretty major flaws. And those flaws are actually the most important part of the whole story. For one thing, it is unbelievably slow. I mean, an enzyme in your body right now could copy this thing in less than a second. QT45, it takes weeks. But you have to remember, on early Earth, there was nothing but time, millions and millions of years. So being slow wasn't a deal breaker.
And second, it's sloppy. It only gets the copy right about 94% of the time. That means it makes a mistake pretty often. Now your first thought is probably, well, that's bad, right? But no, this is where it gets amazing. This is the most beautiful part of the whole discovery. Because what do we call those mistakes in genetics? We call them variations. Every mistake creates a slightly different version of the molecule. And just think, what if one of those mistakes makes a version that's a tiny bit better at copying itself, or a little more stable? Well, then that one will start to make more copies than the others. And that right there is the very beginning of natural selection. This isn't a bug. It's the feature that kickstarts the entire engine of evolution.
So you see, this isn't just about solving some obscure chemical puzzle. This is about witnessing, in a lab, one of the most incredible moments in the history of our planet.
So what's the big takeaway from all this? 3 things. 1. We now know for sure that a self-replicating RNA molecule can exist. It's not just a theory anymore. 2. This makes that whole RNA world idea look a lot more plausible. And 3, and this is the big one, it shows us how simple imperfection, just making mistakes, could be the very thing that got the ball rolling for all of evolution.
Look, we'll probably never know the exact recipe that created life on Earth. But a discovery like this, it gives us a clear, believable path. It shows us a moment where plain old chemistry, through nothing but trial and error, started to act alive. It's not a story about a single lightning bolt or some kind of miracle. It's just chemistry, slowly and beautifully learning to remember itself.